Welcome to the 24th webinar of the Westlake Proteomics series on circulating proteome. This series is jointly organized by Westlake University, Westlake Laboratory, GOMEX, iMarker Lab, the Proteomic Navigator of the Human Body, CNHuB, Westlake Omics, CoShare, and SciX. We would like to pay a special tribute to the Proteomic Navigator of the Human Body project. It is our great honor tonight to have invited Professor Zhouchen Xuan from KTH Roy Institute of Technology and Dr. Xiaobo Yu from the National Center for Protein Sciences. Also joining us in the panel discussion will be Dr. Shunce Tao from Shanghai Jiao Tong University, Dr. Yin Jiang from the National Center for Protein Sciences, Dr. Chen Yang from the Man Ray, and Dr. Tianan Guo from Westlake University. So Dr. Xuan is a professor in translation proteomics at KTH Roy Institute of Technology and Science Lab in Stockholm, Sweden. His research focuses on circulating blood proteins to map human health and diseases. Zhouchen is affiliated with the Human Protein Atlas, a HuB Council member and chair of the Human Plasma Protein Project. His talk today is the dynamic architecture and molecular phenotypes of the circulating proteome. And Dr. Xiaobo Yu, he is currently the PI of the Proteomics New Technology Group of the National Center for Protein Science. He has presented over 10 projects, including the ones funded by National Precision Medicine. He has published more than 40 papers in the leading journals of proteomic and translational medicine, such as Nature Protocols and many other journals. Also, his papers have been cited over 1,100 times by Nature Methods, Nature Reviews, Microbiology, and Nature Reviews. And he has given oral presentations at international and national congresses from many times. And he's the winner of many awards in the field. His laboratory's research focuses on the early detection, treatment prognosis, and the development of diagnostic reagents of tumors, autoimmune diseases, and infectious diseases by using new technologies and methods such as high-throughput protein microarrays and systems biology. Dr. Yu's talk title today is the utility of circulating proteome for precision medicine. Now, before we move on to the talks, please also allow me to introduce all the panelists. So, Professor Shunce Tao received his PhD from Tsinghua University under the supervision of Professor Jing Chen, a pioneer of microarray technology in China and academician of Chinese Academy of Engineering. Prior to coming to Shanghai Jiao Tong University, he was a postdoctoral fellow in the lab of Dr. Han Zhu, associate professor of Johns Hopkins University, who is well organized for creating the first proteome microarray, and his continuous and high-impact works of protein microarray technologies. Dr. Yin Jiang is focused on liver proteomics research and plays an important role in the major international collaborative project, Human Liver Proteome Project. This is to build a liver proteome reference atlas and an international shared data resource to establish hepatocellular carcinoma proteome molecular classification model and to discover a series of protein markers targets for liver diseases. She has published more than 50 research papers in Nature Hepatology and many other journals. And she has edited and translated 6 academic books or chapters and has been granted 9 national invention patents. Dr. Yang Chen is the chief engineer of Shenzhen Man Ray Biomedical and Electronics Co-Ltd. and is the clinical affairs leader of the in vitro diagnostics division. He is also a member of the laboratory education branch of the Chinese Society for Higher Medical Education and a member of the hepatology group of the Guangdong Medical Association's laboratory medicine branch. He is the editor-in-chief of the blood cell analysis section of the clinical testing equipment book, volume 2, Instruments and Equipment, the editor of the In Vitro Diagnostic Industry Technology textbook, the editorial board member of the Medical Testing Innovation and Entrepreneurship textbook, the editorial board member of the Advances and Applications of Clinical Blood Cell Testing, and the editorial board member of the Biomedical Testing Technology and Application. His main research interests are in vitro diagnostic product design, clinical validation and verification of in vitro diagnostic products, and innovative translation, industrialization of in vitro diagnostic products. Last but not the least, Dr. Tiannan Guo is a tenured associate professor at Westlake University, the director of the iMark lab at Westlake Laboratory, and the founder of Westlake Omics. He has been engaged in the proteomic study and is dedicated to the interpreting biological processes and assisting in the diagnosis and treatment of diseases by integrating biomedical research with artificial intelligence. Without further ado, let's welcome Drs. Zhouchen Xuan and Xiaobo Yu to share their talks.
Okay, yes, I'll start. Okay, thank you very much for this very kind introduction, and I hope you hear me well and you can see my slides well. Yeah, it's a great pleasure to talk about the research that's ongoing, I think, in my laboratory and elsewhere, and really with the focus on looking at proteins in circulating in blood and what they can tell us about human diseases, but also what can it tell us about the human phenotype in general. So I will sort of cover three main areas, and of course these will remain interconnected. I will talk about the universe, so we'll talk about the efforts that are ongoing to measure proteins that circulate in blood. Then I will talk about the utility, meaning how can we capture the information that is stored in these circulating proteins, and how can we address challenges when it comes to the dynamic of these proteins and the individuality of looking at the patterns that these proteins inform us about. And lastly, I will talk a little bit about the translation and touch upon a topic that I've started to work on tremendously when COVID came along, which means can we enable precision phenotyping also outside of the clinic? So at first, and as you may remember from the introduction, I'm part of the Human Protein Atlas project, which has started in Sweden 2003 and first launched the public version of this project in 2005. And I think we're until today, we have really come very far in terms of providing deeper insights into protein expression across human organs, tissues, and cells. And of course, this information is extremely important to understand the basic concepts of human biology, but also I think moving forward is to create knowledge about how can we utilize these proteins to study human health. And as you may recall, and I hope many of you have had spent some time on studying the Protein Atlas portal, is that the primary information that is stored there is looking at cellular locations or tissue locations of proteins. So which means primarily focused on studying proteins that are inside or on the cell surface. So studying the proteins that are outside of the cell, of course, is a little bit more challenging because you don't really have a fixed location which you can use to reference the protein and its relation to its neighboring environment. But of course, what we can do, and this is what we started to do a few years ago, is to understand which proteins should we actually expect to be present outside of the cellular environment. And of course, here we started to look at whether proteins are being actively secreted. So as we know today, there are about 2,500 proteins to which there's genetic information telling us these proteins are about to be secreted. And then, of course, it matters where these proteins will then be sort of sent to. So what is going to be the destiny for these different proteins? And what we sort of studied in a sort of systematic investigation.
Hi, Johan. Excuse me. So are you changing the slides or still use the same slides?
Which slide do you see?
We are watching the Human Protein Atlas from maps to knowledge. So
this
slide
is the
did not change.
Yes, I'm still here.
Okay, good.
I will move on soon. Yes. So what I was just saying is that we know that there are about 800 proteins that are actively secreted to blood. And of course, then the effort is that to characterize these proteins a little bit more in terms of their functionality and in terms of their relationship to human disease. As you see now, the next slide, hopefully. But no, that there are various processes besides the active secretion that causes proteins to appear in the circulation. I mean, we know today that people have detected more than 5,000 proteins in not one single set of samples, but globally. So there must be many more reasons why proteins appear in the circulation. And as you can sort of see depicted to the left-hand side, some of these processes relate, for instance, to cellular turnover. So you have cells that leak, that get destroyed, that sort of end their life cycle, and therefore where do proteins sort of end when the cell sort of bursts? It's partly that they are found in the circulation, but also, of course, proteins may be shedded due to active processes as they are being cleaved off the cellular surface. So again, when you think about the conceptual background of the circulating proteome, there are many different entry points from where these proteins can come from. And of course, you can continue to explore and reference these proteins with regards to the organ which produces these proteins. And I guess many of you know that the liver is the primary organ producing proteins for the circulation, albumin as being the most famous one, I suppose. So we have, of course, a good basic understanding of where proteins might come from. But of course, the challenge increases when we want to understand if a protein comes from a specific organ, if it is being produced by several organs and not just a single one. So if we want to really understand and capture human biology, which is, of course, a multi-organ view on the human phenotype, there are many different things we need to consider. And on the right-hand side, you basically see sort of a depicted diagram, which sort of explains some of these dimensions that I think are important to consider when we want to understand the information that we generate from the circulating proteome. Of course, we have to start with the design of our study. Sometimes we as scientists have an influence in saying we want to have a longitudinal study, we want to have a study with these numbers of individuals, but sometimes we rely on retrospective material, which has been collected probably many years ago, and we have to sort of try to understand, okay, what is the information that we can obtain from these. The second thing, of course, is that we want to understand the proteins. So when the targets of interest, are sort of the main focus, where do these proteins come from? What are the processes that lead to them being found in the circulations? And what are the other traits, the molecular traits about these proteins? For instance, isoforms, post-translation modifications, interactions, and so forth, that lead, to gain deeper information insight into these proteins. The next thing is the sample, and this is unfortunately still so much underrated as a component contributing tremendously to the quality of our data. And that means we need to know when a sample was collected, how it was collected, stored, and processed. Because if we do not have this information at hand, it's very unlikely that we'll be able to have a new biomarker that can be replicated in independent studies, because we need to know any of the possible bias that would influence our information that we generate. So really the hope is in the future that there will be more standardized reporting on how samples have been stored and collected. Even though this as a scientist might be sort of a little bit of a, let's say, boring exercise, it is a very strong factor influencing the data that we generate today. Last but not least, we of course want to understand the human phenotype. So meaning here the component that I called traits. Yes, of course, we want to look at, let's say, a biomarker differentiating a case for control or predicting how a human would respond from taking a drug, if it will be sort of improving or worsening the condition. But there are many factors that influence how an individual responds, for instance, to a disease or to a treatment. And yes, we can collect information in the clinic, such as age, BMI, or the use of medication and other sort of lifestyle-related factors. And of course, now we are getting a better understanding the contribution of genetics on these information that we generate. But last but not least, there's a component which, again, is up unto today's standards, often is forgotten. This is the history of the individual, living a life years before the sample has been taken. Which means, has somebody in the health history, experienced a series of diseases, and that sort of, increases the risk of a person being sort of less well responsive to a treatment, or has a person, kept a very healthy lifestyle without taking any medication. So these differentiators are often still sort of hidden variables we still have not captured to understand. And there unfortunately are still very, very few studies that have been following individuals over their whole life to understand at one point in life has certain, a disease occurred, or let's say a longer term inflammation happened to influence the individual's health status. So one of the sort of initiatives that you heard about me that I'm leading is the Human Plasma Proteome Project. That's together with all of these colleagues you see on the right-hand side. And I'm really happy that Chabu and Shengqie are also here, that helped us, to really gather what has the community out there using proteomics as a sort of general concept been gained to understand from studying proteins in the circulation. So this is really, I think, a very global activity, because I guess we need to have these different perspectives and aspects that people are working on to really sort of get the real picture about what proteins in the circulation actually tell us. So every sort of second year, we've been writing a review about the different status on the number of proteins and the different sort of highlights. That's last time in 2021, of course, we talked a lot about COVID, but we talked also about extracellular vesicles. So I really invite you to sort of take a look at these. And we're currently in the process of preparing for the 2023 version of these reviews, which again will bring in some of the recent sort of advances that have happened during the last 2 years. So really that's sort of the community understanding of what is possible and what should be sort of the things we should watch out for when we engage into plasma protein analysis. Because there are a couple of things that I think are important to highlight. One is that, as you see to the left-hand side, is that, of course, with instrumentation, with procedures being optimized, many more people using these, becoming interested again in studying the proteins in plasma. The analytical depth per se increases tremendously. So in 2017, we knew that about 3,500 proteins could be detected. Well, in just 7 years earlier, it was just around 1,200. So of course, that's a lot due to technology development. But it's not enough to say, okay, with the technology, I can measure more proteins, because as you see in the center, it's only the abundant proteins that you can measure in most of the experiments that you do, right? So that's basically the top left corner of this graph. If you look at the bottom right corner of this graph, these are the proteins that presumably are less abundant, or meaning they're being much more difficult to target, which as a consequence means that you will not be able to find them in as many studies as you are looking at. So which means that the coverage and the number of times we can detect a protein here, particularly in mass spectrometry, is really an important aspect. So we don't have any gaps in the data matrices that we want to investigate. And lastly, when you think about the content, so here, for instance, is a graph differentiating the extracellular vesicles, is shown in blue, and plasma only and shared number of proteins. That, of course, we have additional components in blood that influence the things we observe. And then, of course, we need to understand, why do we have these components in blood, and what does it actually mean for our investigation? Are these leakage products from blood cells? Are these extracellular vesicles, and so forth? So again, we are really at the stage of understanding more about this, and I think there's a continuous effort needed to really disentangle this further. So I mentioned, the circulating proteome, and I mentioned the secreted proteins, and this is again sort of to summarize that. Of course, we can use mass spectrometry, we can use affinity-based proteomics, but all in all, I think the overlap of these technologies will give us sort of the truth and will allow us to move forward, because not mass spectrometry or affinity proteomics alone will be able to solve all the questions and understanding human biology. Because again, then when we think sort of a step back, what is the information that we can learn from studying the circulating proteome? Well, we can learn about the individual, but what are the aspects we can learn? One is, as you see to the left, we can look at organ-specific proteins, which are, as we know from the Protein Atlas work, very, very rare. There are only a few proteins that are unique to just specific organs. Then we have proteins in the center that I introduced you to, which are those that we can technically measure, which can be found, and hopefully they will tell us something about the homeostasis, about the sampling. But I guess what we're really interested in are those, let's say, disease-specific proteins, which, change in relation to a phenotype, for instance, taking medication, becoming sick, or recovering from a disease. So it's this package that I think we need to think about and move forward in our investigations. So again, I come back to this wheel that I showed you in the beginning. So we're really focused on getting the technology in our hands and better, more reliable, more translational. But again, what we want to do is to use this information to learn something about biology. And in this sort of transition, we need to consider all these factors that are sort of depicted in the middle, because the combination of these will actually, help us to eliminate any possible bias that would influence our information. So study design is again also something that people not often have a very sort of high interest in, but it's really that you have a donor-specific information, which is like, let's say, healthy and disease, but it's also the sample-related information and how you actually design your study in terms of randomization. Because if you look at from the left to the right, at the bottom, these images depict that, there are many processes involved when you actually get the sample from the individual into your analysis plate in the clinic or in your laboratory. So We have been working a lot with longitudinal studies because we think there's so much to learn from them. And one of these learnings is sort of shown here. So you have the measurements of two proteins, CD5L and caldesmone-1. So, and at the top row shows you the technical replication of this measurement. So you can see when you measure this protein every third month in a group of 100 individuals, the accuracy is really, really good. What you see at the bottom is, of course, not as sort of clear as the green and blue dots, because what we also did is we sampled 100 individuals every third month for a period of 1 year. And we wanted to see how reliable are these protein measurements that we actually do over this course of 1 year. As you can see to the left-hand side, the bottom row shows it's a very high correlation between the first, the second, third, and fourth sampling, which means that this protein is extremely stable. So it's a very reliable biomarker that allows us to, get confident in terms of when something changes in relation to the baseline value, there's something going on. On the right-hand side, you see that this protein basically has zero correlation between a sample taken from the first and the second, third, and fourth visit, which of course indicates that there are many factors that may influence this, but it makes this protein a very unreliable biomarker. And the reason for this protein fluctuating, as you can see, is due to the fact that it's a part of the smooth muscle, which means that every time a needle penetrates the skin, when you take up a blood sample, you will take up different amounts of this protein, and this is the fluctuation you get. So this is basically the variation you get by just punching a needle into the veins of 100 people 4 times during the period of 1 year. But of course, it has tremendous consequences if you would want to use such a protein as a biomarker. Because what we know, and that's what we see from the same set of 100 individuals, if you would see their global protein profile, these are 700 proteins that we used here, they are very, very stable, which means these confettis that you can see on the left-hand side is basically the 700 protein phenotype from 100 individuals, and that they're very stable, which means over the course of 1 year, their phenotype is very, very constant. You see a couple of individuals moving here and there from time to time, but that can be explained by, for instance, people coming back from vacation, people having infections, people having certain aspects of their life change, which causes them to be more stressful or maybe, not being sort of as healthy in their lifestyles. Another thing that we found in these individuals, and that's depicted to the right-hand side, and if you can see the different color codes, these represent the genotype of these about 15 proteins. So each of these genotypes in this illustration shows very clearly to have a very strong influence on the level of the protein that we measure. So without knowing that this is a genotype-driven protein level, for instance, you would have these 2 individuals that have a very, very low level of this CFH protein. You would deem these, for instance, being either sick, or you would deem that there's something wrong with the sample. But because we did longitudinal measurements, we have consistency in the information that we obtain, which means, okay, we trust the fact that these 2 individuals are much lower. And now that we have genetic information, we know it's basically a genotype-driven feature that reduces the protein levels of these individuals. And we have a couple of more examples here. Actually, it's just one single individual out of 100 for factor 12, and we know from epidemiology that the frequency of this genotype is about 1% in the population. So basically, we replicated what has been known in the literature. And then, of course, you can look at also other features in terms of how do proteins fluctuate over time. Is it because they're increased and decreasing in trend, or are they sort of stay above the baseline or below the baseline of the rest of the population? Or as you can see to the right-hand side, you can look at the coordinated changes of these proteins over time. So again, we have so much more information now at hand that we're trying to understand what the relationship of proteins to each other is, what can they tell us about the phenotype, but we see also that there's some coordination about these phenotypes that we need to further investigate. And I mentioned to Tianan before because he, in a recent paper, talked about a protein called histidine-rich glycoprotein, which we have been studying before in relation to aging. This is a work that, now we've been analyzing about 4,500 samples coming from different cohorts. And we were pretty sure in all of these investigations that this protein is increasing with age, and it's actually also predicting all-cause mortality. And of course, we wanted to understand what the reasons for this are. And what we found is actually that using one antibody, we got a very sort of clean GWAS analysis result, meaning this Manhattan plot, as you can see to the left, which means that there's a single sort of gene influencing the levels of this protein, and we luckily were sure that this is actually a relationship in cis, which means it's actually the HRG gene influencing the protein levels of this protein. But when we used another antibody, we again could replicate that this is the gene influencing the protein levels. As you can see in the center, the relationship between the genotypes for these two antibodies is perpendicular, which means this has a very different relationship. So if we just look at this one specific variant, so proline 186, it's actually a position in this protein which is highly influential, which means it's very, so proline switch to serine is of course a very structural change that occurs from a helix breaker to a turn maker. And it does also affect the local environment, as there's a cysteine bridge and there's a glycosylation site. So at this one amino acid change, we could sort of learn so much about the biology of this protein because it has such a huge impact on its functionality. And then when we looked at the protein in a little bit more detail, we of course figured that, okay, this is not just a single domain protein. It actually has these 6 domains that all have been reported to do different things. Maybe HRG is a specific case, but in our example here, we had one antibody targeting this N2 domain, and we had another antibody targeting the C domain, both which we validated by the genetic association studies. And as I showed to the right-hand side, each of these antibodies give you a different phenotype. They measure the same protein, but the genotype, the information that they provide you with clinically is different, as sort of highlighted by these blue and red marks here on the table. So again, you measure the same protein in the same assay in thousands of individuals using 2 different reagents, or it may be, you can translate it, you look at 2 different peptides, and they may tell you a completely different story. So I think, again, these are examples that I think we need to think more about in terms of learning more about human biology. Because we know, of course, that we can do all these decomposition analysis to see what is the influence of genetics. And we know this is around, like in this study that we've done here, about 15%. So I mean that there's an added sort of component of lifestyle and environment, and maybe also people taking medication. As you can see to the right-hand side, this is again from this study with these 100 individuals. When people, for instance, lose weight, this is the example on the bottom left under B, you will get a change in protein profiles for a tremendous number of proteins, as you would get for this other example, where a person in one visit has actually come back from vacation and with an infection. So, you know that there's a quite strong response to these environmental changes. And then, of course, you can sort of put all the other omics spaces into this, and I'm not going to talk about this in more detail, I guess, due to time. But again, what I want to say is that we can use this information that we now generate from the proteomics and other omics data set to do 2 things. Okay, one, we can sort of confirm, okay, this is the clinical association to, let's say, this variable, or we can just say, well, let the data speak to us and then define a molecular phenotype that is based on the data itself and not on the silos that we impose on the data because we have some prior knowledge. And this is sort of one of these sort of soft clustering activities that we did in the context of type 2 diabetes. And here we could show, as depicted to the right-hand side, that these molecular phenotypes that we are creating are actually consistent if you look at longitudinal sampling. Again, also the effect of medication is something we need to further investigate. Here's the use of metformin. On the sort of right-hand side, you can see the phenotype differences of the individuals that we are measuring. And on the x-axis, you see the actual change longitudinally in terms of the effect of the drug on these individuals. And here, for instance, GDF15 is a well-known biomarker for metformin use. Very nicely confirms that this has a very strong impact on the individuality, but also on longitudinal behavior. So during the last part of my talk, and I hope I have another 5-6 minutes, just checking. No complaints?
Yes, please go ahead.
Thank you.
Yeah.
Yes, thank you. So during the last part of a couple of minutes, I wanted to talk about this, new way of trying to understand the human phenotype, and really imposed by the COVID pandemic, but also by this article that came out in Nature Medicine at the end of during 2020, which sort of says, okay, what is really the future about doing precision medicine or here clinical trials, as sort of the authors have been sort of focusing on. And is it really that we need to involve the clinician, that the patient actually has to come to the hospital at a given time point, and has to travel and has to make all these live arrangements? Or are there other ways that we can actually engage individuals into studying the human phenotype? Can we do this in a more controlled manner? Can we do this more frequently? And can we, for the COVID, situation, can we do this in a pandemic where, for instance, people don't really want to travel? So this is really sort of a milestone for us in terms of rethinking the way that we want to approach, studying the human phenotype. Because, as you know, and this is another study that I've been involved in with colleagues at Charité in Berlin, of course, can have this excellent sort of a description of human health in the population when they come to the clinic. So here we did a study on 150 individuals that enrolled into the clinic, and then really were categorized by these different tests that were available at that time. So what really this showed is that you can categorize individuals, and what we're trying to do in this exercise to differentiate those individuals that had responded, or that actually in our case survived the COVID-19 treatment, from those that actually were more severely ill. So you can do all these measurements in a really defined clinical environment. But how can we go outside of the clinic? Well, what we have been doing is to engage into home sampling, which means we're sending people packages that contain all the material that they need to leave a drop of blood. And then they send the drop of blood back to us in the lab, where we can perform the detailed analysis. So, okay, it's like a Theranos that people have been, criticizing, wondering, and being concerned about. But really what this actually is, it is the possibility for future medicine. So we've been using this device from a company here in Sweden called Capitana, and the smart thing about their concept is that you use a microfluidic chamber to control the amount of material that you actually collect. So at the end of the day, we have a little disc, which is sort of depicted on this side, which contains exactly 10 microliter. And the smart thing about this is that you can just use this disc, and you don't have to be concerned about the amount of material you put into your system, because if you would go sort of the classical way of punching out a different area from a sort of piece of paper, you are not sure whether the amount of material you have is actually equal to the amount of material you will obtain from a second person. So we've been doing this in the pandemic, really first wave, when I think the whole world gone crazy. We've sent this out to 2,000 people in Stockholm using random addresses. We got a fantastic participation of 55%, so more than 1,100 people provided us with blood and with a little bit of information about themselves. And, to the surprise of many, but really as it showed later, that about more than 10% of the individuals had actually been exposed to the virus in the first wave of the pandemic. And also, I think what people later showed, that the Alpha variant, that were about 30% of the individuals who were asymptomatic. So they didn't even know that they have been exposed to the virus. So again, we demonstrated this by going directly from the lab to the population, that you can do these type of measurements. But of course, serology is fantastic, but we wanted to go a next step further. And as you can see to the bottom, we're working on several studies that I don't have time to talk about, where we actually want to do deeper phenotyping. So we use proteomics methods. 2 more minutes. Proteomic methods to really disentangle this. So we can measure these variability across these different samples, because dried blood spots, people may be concerned about the quality. We know that the quality can be very good. We can measure longitudinal profiles. We can see very interesting trends when it comes to the, for instance, this is an individual under recovery. And we can also, look at the phenotypes associated to the different stages of the infection. We can also look at the relationship between the proteins, because that may, again, tell us something about the functionality of the phenotype. So then we can go even deeper, because new technologies become available. And this is something we've been doing with Olink, because it was, just down the road here in Sweden. So now we know that we can actually measure more than 1,000 proteins in just these 10 microliter of dried blood, which has been collected in the population, sent back to us into the lab for us to do this analysis. So we know that you can actually engage individuals who are not trained to do this type of exercise, and you can really get highly precise and fantastic information. So what I think the next step is, and this is also something we've been demonstrating, is you can actually even go smaller. So you can actually use some sort of a microneedle system here to collect, for instance, interstitial fluid. So it's the fluid between the different cells. And you can actually in here very nicely measure if an individual has been infected with the SARS-CoV-2 virus, the concentration of antibody is about 100 times lower than in blood. So you need to use more advanced methods to do these type of investigations. But another thing, and that's sort of my last slide, you can also develop these on-chip systems to do a little bit more at the time point of sampling. So here we've been using a sort of a chip to actually do blood preparation. And then on this preparatory device, we're actually doing capturing of the proteins from the freshly prepared blood to measure some of the relevant proteins that are interesting, for instance, in the context of inflammation and infection. So we know that technology is not only increasing the number of proteins, number of samples we want to measure, but it's also increasing the possibilities to do something between the arm of the donor and the instrument, which is, for instance, using microsampling devices for future studies. So with that, I hope I didn't go too much over time, and many people I want to thank, and of course, happy to take any questions now or when the schedule fits. Thank you very much.
Thank you. Hi, Joshua, can you hear me?
Yes.
I have a burning question. So when you talk about DBS, you can sample it at home.
Yes.
For that type of sample, which method do you use to measure the proteins, with antibody or Olink?
Yeah, we use all sorts of different methods. So, I mean, Mike Snyder had a paper recently out in Nature Engineering. I think he showed, you can do metabolomics, you can do all sorts of things. So it's really about, of course, adapting the device or the sample that you get from the device with the method. So we've been using Olink because it's for us the fastest way, and it consumes only very little amount of sample. And we've been actually doing this, from samples we first characterized with serology, which also I think works fantastically. And now been, expanding this because, of course, at some point you need to, think about what is the phenotype that you're interested in. Is it inflammation? Is it cardiometabolic? So that's, I think that the door is wide open, and I'm sure mass spectrometry will be a player there too. It's just something that we had at hand. We used, also Luminex, we used Quanterix, and I'm sure, targeted or exploratory methods will be equally informative. So it's just what I think is important is to really get the quality of the samples as high as possible, because the variance you may get from having different people doing the same thing can be, exaggerating the differences in the molecular levels you want to measure.
Yeah,
so
true.
thinking about, reference proteins or similar things is an important exercise.
Yeah, true. I noticed you are using an envelope to ship
Yes.
the sample to the small tip to the lab. So
Yeah.
how long does this take? Do you have a quality control? Let's say your mail, I don't know how that would take in your country. It's several hours or one day. Will the protein be somehow modified or degraded?
So I think the important thing is, so this device, as you can see, it folds together, which means you have a first layer of seal, and then you put it into a desiccator. So you want it to be dry, and that takes about, let's say 1 or 2 hours. Ideally, it's overnight. Then it's dry, and then as soon as water has disappeared, it's much easier to know. Okay, the quality won't change. Maybe it will change 5-10 months down the road, but not within a week or a month.
You mean at the room
temperature?
You mean at room temperature or in the fridge?
It's room temperature. Yes.
Ah, cool.
Because as soon as water has disappeared, what will happen? So then, of course, if you keep it at room temperature, which will be when it's been sampled, maybe in the summer or winter, the difference will be when it's in the post office or in the in the big let's say processing unit. But we don't think that there's a huge effect from that. The key really is that the quality of the sample, and this is the nice thing with this device, it controls the flow of the liquid into this measurement component. So that sort of gives you an initial QC. Then you have a visual inspection. You can look at, as sort of I try to depict down here, you can actually see how red the dot is that the person has collected. And then you can eject this little device. I have one actually here. So you have a little disc here, and you can take a tweezer to take it out. So then you have something already, formed in your hand that you put in a microtiter plate, and everything is done by robotics afterwards. So it's very handy in that sense too. And then you can control these conditions, of course. What of course we've been thinking about is to label these cards with a barcode, and then you can develop an app where a person takes a picture, and then you have, you know, much more controlled sort of processing time and time point of collection, so forth. So
very cool. So Sensor, do you have any?
Jochen.
Hi.
Yeah, hi. Long time no see. Okay.
Yeah.
So I also have a follow-up question about the micro sampling. I think it's cool. You can control the volume, right, very precisely, even you're sampling at home. So the question is like, is there spatial like a treatment of there should be a filter paper, right, underneath? Is there like any spatial treatment for the paper, like some kind of chemical?
Yeah, I mean, the whether or not the paper is treated in a special way, we have to probably, you know, ask the company. I think that they're trying to use this as standardized material as possible, because you know, otherwise they would have to, you know, get all the accreditation for coming up with a new material. But I don't have the details. I guess the key is, again, if I maybe can manage to start the video, is that you have this landing pad down here, which basically sucks out the liquid.
Yes. Yes.
And then you basically create this space here in between these two discs, which is exactly the 10 microliter. And then I guess it's just, you know, if you play a little bit, you know, at home with some of those, you know, devices, you can actually, you know, transfer liquid. So that's, I guess, the concept. But of course, you know, you need to think about the flow of red blood cells. You may have initial coagulation, and also maybe I can mention that this white landing pad up here, that's hydrophobic, which basically means that the drop will actually sit above this little sort of outlet. Okay.
So another like a small question is like, what's the price for this one?
I mean, it depends. It's I think people estimate that using remote sampling is about, what is it, 100 times cheaper than doing it clinically, because I mean, the cost is not only this device, the cost is also, you know, the nurse, the environment in the clinic. It's logistics around that you need to put into the calculation, because sort of the sheer cost of plastic, you know, wear or syringe and things like that, I guess that's just, you know, a number of volume. So, but if you put, and I can share, I guess, some of these conceptual studies with you if you want, where they actually, you know, try to really calculate the exact price difference. But I think
why I'm asking, why I'm asking this question is like there's maybe there's one possible application for this kind of device. Like when we do some kind of study, right, we always want to collect a lot of sample. Sometimes if we can collect sample at home, that will be pretty cool.
Yes.
Say if I have a study, I want to collect sample from 500 people. Then we can also use this kind of like a microchip device, collect sample, just take all sample together, then do the analysis. Right? That's another way. So in this scenario, the cost of the sampling device is we should take it into consideration, right?
Yeah, I think the idea, I mean, we've been now doing a couple of studies, you know, with cohorts where people have been, you know, invited to collect samples. And the ideal case is that you do the first sampling, let's say, with a nurse. The nurse explains the person how to do it. And then they get a package with them to their home, and then maybe you can, every 2 weeks you give a drop of blood. And I mean, I've been doing this also myself because I wanted to know how good it works. And it's really fantastic. So I mean, you know, there are other like devices like Mitra and other companies that develop these systems. I mean, I've been collaborating with the co-founders of this company, so that of course made it easier to explore the possibilities.
Okay, that's cool. Yeah, thank you.
Are there more questions, or what's the plan?
Hello, I have a question.
Yes.
I want to know whether you have been concerned about the protein composition of blood samples changed over time during storage. Sometimes we will to analyze the proteomic on blood samples taken at different follow-up times, such as the fresh collect the samples and the samples stored for 2 years or 5 years. What I want to know, how about to consider about this variation?
Exactly. I mean, it's a very good point. I think there are a couple of papers that show that with various methods, but our understanding is that the age of the sample is as influential as the age of the person giving the sample. So these two sort of age factors are really, I think, interdependent, but important to be considered, right? Because even if you have mild degradation, you know, it might just going into the wrong direction of the measurement differences you want to achieve, right? So I think having the time point as a factor is important. What is even stronger as a factor is the location where the samples are collected. So if you do a multi-center study, the first two components, if you would display your omics feature, and you will get sort of a map of different, you know, islands from your PCA or UMAP or TSNE, what you will see is the location where the samples are coming from. And it may be, you know, lifestyle, genetics, it may be sample handling, but again, you know, it's these factors that we need to bring into our understanding because they will have a driving, you know, influence on the phenotype that we measure.
Yeah, just to have an idea. So since you are leading this human plasma proteome project, are you thinking about to have some standard samples you can ship to every center? And I don't know who this huge amount of blood, maybe a mixture of different use that
I think this is how it actually started, you know, when Gil Oman and others, I think it is around 2003 or 4 or even 5, you know, they had this, I think, Nature Biotech paper where they exactly tried to do this and, you know, prepare one sample and send it to different labs. And it didn't work. I think because the influence of the sample material itself, because it's not, you know, it's not a sort of a static material. It really, you know, things change and maybe how people thaw the samples and how people, you know, treat the samples when it arrives. So it's what people have been thinking about a lot. I guess genetics, from my understanding, can be a very good denominator nowadays, you know, to understand these things. But again, the quality of the material and the influence of, you know, pre-analytical factors need to be understood much more. And of course, yeah, standard values is always important because how do you know this is the absolute truth, right? So what is the reference that you say, you know, this is the albumin level you should expect in a sample prepared from blood? But there are so many factors even influencing that. I mean, you know, it might be in certain ethnic populations, you may just have, depending on what variant you measure, different levels that you obtain. So I think people will work on this, but it's actually more complicated, I think.
Yeah. I know the story about the Nature Biotech paper organized by HuB early 2000s. I think there's a pretty mixture, very simple sample. And the main issue is about the data analysis, I think. Some people, I think from ISB, they collect all the data and analyze them particularly, that is much better. And I think during my postdoc in Zurich, Rudy's team organized a study.
The HeLa study,
Lens real HeLa cell, and sent to different labs for SWOT analysis. And then this much more consistent compared to 20 years ago or 15 years ago. So I think there's a still chance. If you want to move to clinic, then I don't know, different country, different authorities to check the accuracy, the reliability, stability, robustness, and so on. So we ask the same question. You can publish a paper, convince reviewers, but how to convince the authorities?
Yes, no, I agree, and I think you know, in one way there's also an opportunity now that we have so much more data from a measurement, right? Can we use the data as sort of an anchor point, right? Rather than say this has to be this value, but let's say if we define certain coordinates in the data that we have, you know, and say this has to be the pattern we want to achieve, the relationship and things like that, and then that will be used as the anchor point for the sample. I guess, you know, people who are working on this, and it's of course continuous question to take the next step into the clinic, right?
I also collaborating with Claudia.
Yeah.
Yeah, I talked to her during Cancun
Yes.
HuB meeting. Yeah, we were talking about to compare the data generated by mass spec by her O-link or other antibody-based method, and to
Yes.
see what's the similarity and difference between different data sets.
Yeah.
Probably another time we can talk together.
Yes.
See what we can.
And as you know, UK Biobank is, for instance, spearheading one of those initiatives. And I think maybe you saw Chris Whelan talk, when, of course, now they have done the first wave with O-link, and now they bring, SEER and other technologies, because, they also understood it's great to have, 50,000 or 60,000 individuals with, I don't know, 1,500, 3,000 proteins, but it's still, biology is much more, complex than just that. So I think we will, I guess, we as the community will have a great opportunity to learn from that. So I think it's going to be fantastic.
Yeah. Sabo?
Yeah, I have a question. Yeah, thank you for your very nice presentation. Just I saw the number of views for your presentation. There almost have 1,500 people now, have viewed your presentations, so it's very good to know. So you already have fans, many fans in China. So hopefully, this is the start, and we will have much more opportunities to communicate, between your team and our teams in China, So one question, for your study, also for a paper published by Tiannan, for the study of the association between the genetics and the changes of serum proteomics, So I saw you did many samples, for these kind of studies, and to identify the linkages between the pQTL and the change of proteins. One question is how we can validate. First, how we can validate the finding. The second is what is the functional consequence of this kind of linkage?
Yeah, validation is often nowadays done the way you do sort of a lookup, or you have an additional cohort where you have these, these references. But I guess people are understanding, many of these initial studies have been focused on, let's say, the European descendants. So of course we need to, expand this, and I think that's also great. The work that Tiannan has, shown. We need to expand it, because biology is not just, looking at one picking up population. It's sort of the global population, and that will allow us to understand also what you mentioned, the consequences. So a validation, of course, of these observations is something that people will have to work on, especially, let's say, finding functional assays to actually show what does a variant influence in a protein function. Of course, you have some, let's say, very sort of strong effects of some variants. So you have truncation, or really sort of changing the function of the protein or turnover of an enzyme but by a single point mutation. But then you have maybe more subtle, a wider distribution of these variants, and then you basically have to look at the system globally. So it's not just one mutation; it's the mixture of these relationships that will be important. So let's say for this example here, where we have HRG, and we know it's a multi-component protein. Maybe it's a bad example because it does so many different things depending on which of the domains you are addressing. But it gives you a rise to that. Okay, if you have a mutation in this part, maybe coagulation will be faster or slower. If you have a mutation in this part, maybe wound healing will be faster and slower. So I guess it's really a systems biology problem that I think we need to address. And of course, I think we're now in a phase where we're learning about this genetic control, and we need to translate it into functional assays. So there's this group on studying common diseases, which basically tries to put mechanisms and all these things together, medicine, so you can actually get a systems understanding rather than say, okay, you have this protein, it phosphorylates or it dephosphorylates, that's it. Well, maybe it's not, right? Because the protein you may phosphorylate may also be under the pressure of having certain variants that are easier to phosphorylate or easier to dephosphorylate. So I guess my answer is it's a complex systems biology problem.
Okay, great, thank you.
Okay,
Okay.
thank you. Okay.
Go ahead. Go to next talk.
Okay, thank you very much. I'll stop sharing my slides then, right?
Next, we're having Dr. Xiaobo Yu from Beijing National Centers for Protein Sciences.
丽琴可以看到我的这个 presentation 吗？
现在还不行。
我刚才应该是共享了吧？
好像可以了。
现在
共享，
可以了。
现在可以了 ，OK 是吧？OK。
Not in full screen.
OK， 现在是个 full screen 了，对吧
Yeah, not in presentation mode.
对，那个我就用中文了，因为我们这个主要面向中国的一个观众所以说今天也是这个通过天南来给我们的一个命题，就是什么是循环蛋白质组然后通过Jan教授，他给了一个全貌式的一个工作一个介绍，并且是他在这方面做出了非常精彩的工作。在我这个 presentation 里面，我想可能我们这个注重的点可能会更偏向于这个如何把我们这个蛋白质组群的研究能够向临床真正能进入转化。因为我们大家现在都在说精准医疗，但什么是精准医疗？蛋白质组是不是真正能精准医疗？其实这个里面是有一个非常浪的一个非常长的一个 story 在里面，所以我这里面今天主要是给大家介绍一下我在这方面的一些思考那么首先介绍背景，我们都知道这个21世纪人类生存发展的最大的挑战就是人类的健康问题，对吧？包括这个现在癌症的发生人数每年也不断上升，包括新冠肺炎，那么也是给我们人类给了一个非常好的一个教训。那么到现在呢，我们应该是说从整个的社会的各个产业来说，那么我们大家非常关注这个精准医疗这一块，所以它也是我们医学发展的一个方向，也是我们国家政策以及行业投资它的一个热点。那么对于这个精准医学呢，我们知道它最开始领先的是它这个基因组学，那么现在呢，基因组学已经出现了大量的很好的其实成功的案例，可以供我们蛋白质组学来参考。那第一个基因组学它用的一个窗口就是癌症早筛的产品，包括就是检测这些 cDNA mutation because 或者说检测一些甲基化的一个方法。那么现在这个很多的企业现在已经拿到一些临床注册证，或者是用作泛癌，或者是用作肝癌那么这是一个基因组学它的一个应用的一个重要的方向。另外一个基因组学应用的一个重要方向就是伴随诊断。那么在这个里面，其中一个比较典型的例子就是 Foundation 1 CDX， 它有324个基因，对不对？然后 MSK 的 Impact 它是468个基因，它都是用来检测临床针对某基因特定药的场景。当然这里面还有一大部分这个基因它是检测单个基因，也同样是针对单个这个 mutation 的它的一些临床靶向药的情况。那么这个是整个基因组学它应用于精准医疗的它的两个很重要的一个应用场景。那么我们知道这个基因组学呢，相对来说基因来讲，那么我们人蛋白质它是更加的可以反映我们人体生命周期的变化。假如说这个 genetic changes 它反映出我们先天的信息，但是我们胎儿生长发育到衰老，在这个过程中，我们人体可能会产生各种各样的这个疾病，包括肿瘤致免或者 COVID-19 我们会接受的相应的治疗，对不对？然后我们这个治疗之后，我们还会监测的复发。其实在这整个这个过程里面，其实我们知道我们蛋白质组学的在里面，其实我们完全是可以发挥更重要的作用。在这里面我们非常关心几个问题，第一个就是内在的机制是什么，第二个就是说我们是否可以解析我们疾病的密码，就是我们找到的标注物，可以开展精准的检测。那其次呢，就是说我们是否找到治疗的靶点，开发新鲜的靶向药物。所以说通过这个图来说，我们蛋白质组学理论上是我们可以在整个的精准医疗可以发挥更大重要的作用。 那么在整个蛋白质组学呢，我们临床有各种各样的临床样本那么血液这一块呢，基因组学其实在作为基因组学，它作为参考来说，它也主要有2种，一种是癌症的早筛和早期检测，在这里面主要还是以这个液态活检为主，以血液和体液为主那么从这个角度来考呢，考虑到作为蛋白质组作为癌症的一些早期检测的工具，它一定也是血液最为理想的一个临床样本。那么其实作为组织来说呢，那么基因组相对来说用这个组织样本更多的是用于这个临床的一些靶向药物的一些选择那这里面呢更多是侧重于这晚期的癌症。所以为什么选择血液呢？就是稍微解释一下，也是切合今天这个合作的一个命题，就是 CirculatingProteome。就是第一呢，血液包含了非常丰富的生理和病理的信息，易于获得并且基于侵略性。还有一个很重要的问题就是说，血液中的蛋白的标志物，它一旦我们进行验证，可以非常容易转化到我们临床的现有的平台。那么基于这个血液系统，就像我们这个今天的主题 CirculatingProteome， 那么血液里面其实它是一个相对封闭的，我们可以把它看作一个封闭的系统。那么优恩也解释了展示一个非常好的原理图。在这里面呢，我有凝血系统、炎症因子系统、补体系统、代谢系统、细胞骨架、免疫系统，还有很多一些细胞死亡或者这个一些组织损伤释放出去类蛋白。那这里面构建了我们整个一个相对的封闭系统。所以对于我们团队了呢，我们就把这个基于血液的血管蛋白质组做一个系统，我们采用系统生学的方法，结合我们以蛋白质芯片为主的这个蛋白质组学，开展这些系统的那么精准一些相关的研究。所以在6月后的他在这个之前的这个 blood 在这个clinical chemistry， 他进一步提出了这个问题，血液是人体和健康提供的一个窗口。那么在2005年呢，其实在美国癌症早期联盟已经启动了一系列的项目，开展一些这个基于血液、体液和组织方面开展癌症的早检测。在这个里面呢，其实也取得了很大的一个成绩，包括 FDA 已经批准了8个临床作证，CLIA 已经批准了19个临床检测试剂。所以说在国外的进展，蛋白质精准医疗它的一个导向其实最近几年发展非常快的。那么接下去举个例子，第一是肺癌的基于多个自身抗体的检测试剂现在已经批准了。那么国内现在也有个试剂叫凯保罗，那么现在已经开始报物价了。那么这个是检测这个7个自身抗体，它是可以在这个肺癌这个组织它这个直径小于2个纳米的时候就可以发现一个早期的肺癌。那么第二个例子就是乳腺癌的检测试剂，乳腺检测试剂这是美国当时赵世磊贝尔领导的实验室发现的，其中包含了一部分的自身抗体，还有一部分的蛋白标志物。这个里面它也可以达到非常好的一个灵敏度和特异性，尤其是对于手术的患者，它可以进行一个风险的一个评估。那么最后一个呢，就是胰腺癌的早期诊断的蛋白质组学的一个检测芯片，这个是2021年8月3号。那么美国 FDA 批准可以用的临床实验检测，这个也是他们对血液中的一些这个蛋白的标注进行联合检测，并且加上人工智能算法，而这个是非常契合我和生测我们整个研究的这个平面芯片的方向。而在美国，它这个批准用临床收费是非常非常高的，那么检测一个样本要$995，所以这个是这也证明了这个国际上对于基于蛋白质组的精准医学来说，其实发展是非常快的。当然，对于蛋白质组学成果怎么去临床转化，之前呢，我们也举办了一个研讨会，当时我们也是跟在座的各位老师，我们进行了很多的探讨。其实蛋白质组学往临床转化，其实难度还是非常大的。那么第一呢，就是大规模的一定要有高度稳定性、重复性、灵敏度非常高的检测技术，尤其是定量的检测技术。那么第二呢，就是说只有这个检测技术是不行的，我们要考虑到这个技术，我们的 data 要往临床转化该怎么办？是否那个网这平台符合临床转化的一个标准？所以第二个我们面临的问题就是说，如何建立临床转化的技术体系的问题。那么我们有了这个很好的技术平台，我们接下来就面临怎么去选择标注，怎么去规范我们的研究的策略。那么再往下呢，就是说我们有好的标注，接下来我们就是要吸引一些国家的项目和企业的资本来投入，因为真正的往临床转化它的投入非常大，包括我们跟与产学研一起来结合，推动我们的行业标准还有行业指南进行学术科普，那么按照临床的标准开发出临床的审验试剂，注册和报批，最后大系统一起来推广，所以这个过程还是蛮长的。那么通过参考这个基因组学来说呢，上一个图，上一个图就这个里面呢，我就把这个蛋白质组学的转化体系跟基因组学的对比，因为基因组相对比较成熟嘛。第一个最大的特点就是基因测序的话，它是可以实现绝对定量的。那包括就是说大规模检测有2代、3代测序，那么中间的这个 size 的 number 的10到1000个，我们有基因芯片，然后多指标的有数字 PCR、多重 PCR，那么单指标最终有 PCR 还有 POCT， 那么蛋白质组学呢，我们在大规模检测这一块，目前主要还依赖于质谱和高密度蛋白芯片相 O-link。这个我们的检测的结果是相对定量的，所以从相对定量这个结果来说的，如果我们临床应用的话，很难达到临床的标准。所以这个时候，我们发现有用的标注，我们一定要想办法要把它做成绝对定量，而且要有一定的相应的参考品。尤其是现在我们临床已经相对比较成熟的流式芯片、单色芯片、POCT， 那么如果说指标更少，那么就是1到3个指标，我们可以落到结合我们的临床的化学分乖，可以迅速的帮我标注推向临床。对吧？这个是我们蛋白质组学临床转化的一个体系。那么首先对于我们来讲呢，我们第一点也是，我们第一点就是打磨我们的技术，一定要高度稳定的蛋白质组学的一个获取数据的技术。那么我们团队主要是聚焦在蛋白芯片这一块。那么我们回国之后呢，我们也是一直来就是打磨我们的技术，包括我们构建人类的蛋白质组芯片，包括肺结核的，还有病毒的，还有新冠肺炎的。那么以前呢，以前内容就更加注重数据检测蛋白的数目。那么到现在呢，我们也实现了一个变化，更加注重于蛋白数据的质量尤其是定量能力。那么通过大规模检测的，那么那个蛋白质组学技术，我们的特点是什么呢？就可以从蛋白质全方位来检测我们靶标这个疾病患者的体内的信息。那么这是我们新冠病毒的一个例子，我们打造这个芯片可以获得新冠病毒全部的针对线性表位的，还有一些空间表位的它的病毒的一个抗体。那么这是我们的技术。那么这是我们2000年做的抽取新冠病毒的它的序列信息，然后设计成高密度的端芯片，以及一些关键的一些蛋白作为探针做成一个组学芯片，开展我们临床的应用。所以它的特点就是说，我们一批就可以获取大批量的这种高质量的数据，包括 IgM、IgG、IgA 的信息。这个横向是随时间点变化，我们可以看到每一种抗体的变化水平是非常非常大的。那么我们检测这个之后呢，我们就可以进一步精确到每一个新冠免疫抗体的表位，我们把每一个蛋白它的表位信息用这个标注的这种全部描绘出来。那么这些信息有什么用呢？那么第一就是我们可以找到预测患者死亡的一个标注分子。那么通过这个呢，我们检测新冠死亡和存活患者的，我们来解析了新冠病毒危重症患者人体生存和死亡的新冠病毒的它的抗体的表达规律。我们发现死亡组更多与这个结构蛋白相关联，那么存活下来那是非结构蛋白关联。那么这个结果呢，我们进一步在不同的队列里面进行的验证。那么我们可以用我们的标注，及时的可以为临床救治提供预警。那么这个是我们筛选了大量的这个新冠病毒的一些重组抗体，我们找到了可以非常好的综合性抗体，对于活病毒的抑制力也可以达到99%所以这个是筛选中药物的一个例子。那么我们有一个好的技术平台呢，接下来就是我们怎么打造我们临床蛋白质组学的转化体系。那首先第一点呢，就是我们瞄准了，我们有这个体系了，我们瞄准我们可以国家这个存在的一些重大的一些生物安全和精准医学的问题，我们开展大规模的蛋白质组学的检测。同时我们还有一个数据库的一个平台对于验证的标注部分呢，我们在小型的芯片上，在不同的队里面进行验证对于验证的标注，我们迅速转移到我们临床的平台来往临床进行推广。那么通过这个模式呢，这个是我们一个软性的平台，那么都是血液中的一些抗体和蛋白标注的数据库那么有一个一软一硬的平台呢，接下来我们就可以开展一系列应用了。那么接下来我就快一点讲，因为今天时间可能也比较紧。那么第一个就是诊断标注的例子诊断标注，这就是我们在2017年我们做的一个银屑病的一个例子，它是号称不死的癌症，中国患病人数650万，其中最大的一个问题就是说，得了这个 UP 选之后，这个银屑病它是很难去治愈的，而且随着我们人体年龄增大呀，那么人体的免疫力越差，那么这个疾病它就会越来越严重。所以当时我们接手是没有任何的分子标注的所以通过我们这个蛋白质组学的芯片，我们全部系统的筛选了健康对照以及治疗前和治疗后的那么银屑患者的人的血清。那么我可以发现红色的是升高的蛋白，绿色的是表达降低的蛋白。那么在我们这结果里呢，我们也通过我们蛋白芯片也跟质谱来进行了对比。那么 Y 轴是蛋白丰度的一个分布，我们可以看到就是说质谱相对来说更好的检测这种高丰度和中丰蛋白。对于芯片来讲，由于采用是免疫法，可以更好的检测它低丰度的一些标注分子还有炎症因子。那接下来呢，就是说通过我们以血位系统，我们解析了那么对于银屑糜烂者他患者体内还变化的这些信号通路，包括补体凝血那么变化的显著性蛋白，我们用红色来进行了表示。那么同时呢，我们也探索这里面是否有诊断有价值的标注分子。我们首先进行了聚类分析，可以很好的把健康种、营养组进行区分开。 ，PCA 组呢可以同样达到同样结果，说明这里面一定是有很好的一个标注在里面。接下来我们就跟临床数据进行了相关分析，左面是临床数据，右面是我们鉴定的标注分子。通过这个关联分析，我们就找到了跟临床金标准相关的一些蛋白，包括 PS3， 包括 CR22， 那么 PASI 的是 IR12B。那对于这些标注分子呢，我们在图例里面又进行了队列，同时我们就加入了相关的其他皮肤上的对照。那么对于红色的这个银屑病呢，我们可以达到非常好的一个检测效果，并且是它的相对于不同的这个相关的皮肤病来说，它的灵敏度和特异性都是可以达到70%，特异性可以达到100%，所以这个效果是非常非常好的。那么对于早期呢，上面是早期的结果，那么 AUC 也可以同样可以达到 0.8 以上这个左右这个级别所以这个时候我们找到一个非常好的标注。另外就跟治疗性它有非常显著的关联性。那么我们拿到了2个队列 treatment 1 treatment 2，那么 N2 是治疗无效的，2是治疗有效的。我们看到这个标注在治疗无效的人群，这个标注是没有任何变化的变化度非常小那么对于这个治疗有效的人群，它的标注下降水平是非常大的。那么右面是做一个 R 和 N2 的一个 box plot 表示，我们可以看 N2 组，那么对于2组的下降是非常明显的，而且这个对于有效和无效它具有非常好的区分能力 ，AUC 可以达到 0.75， 而且在2个队列里面我们都可以达到一个同样的结果，所以这个工作我们现在也继续写了。那么接下来呢，就是说我们达到了一个非常好的一个效果，那接下来我们就涉及到临床转化的问题，所以这个我们就跟这个达瑞生物和广东省中医院，我们拿到了一个广东省转化移动专项。那么通过跟我们擅长做体外诊断试剂开发的企业，我们联合开发了相应的标准品，还有体外诊断试剂。那么现在也是启动临床注册了。那通过这个结果呢，我们是想就是说我们是真正实现产学研的这个有机的一个结合，那么进一步推动那么疾病的临床诊断进入这个分子定量的时代，让更多的患者来进行受益。那这是第一个例子。第二个例子也是一个稍微比较有难度的一个例子，就是通过血液蛋白质组怎么进行分子分析。我们知道就是说血液蛋白组跟组织来讲，鉴定蛋白数目要小于10倍到20倍以上，就是我们蛋白100以及可能是500到1000个，但基于这些蛋白我们来怎么进行分子分析。所以针对这个例子，我们选择了一个临床抑制性特别高的一个白塞症做一个这个实例，因为它是第一，它这个患者就有高度的一致性，同时呢，它的这个疾病受累可以受累人体很多个器官，包括皮肤包括神经包括关节可以说这个器官。那么基于这种抑制性的疾病，那我们怎么通过血液蛋白组怎么来解决这个问题？那左面是我们整个的一个实验设计，那么右上角呢，这是我们同样的一个检测的一个结果，进行了一个深度蛋白质组织的检测，通过结合我们 DMS 和抗体芯片，下面是抗体芯片检测的一个结果。我们拿着这个几何结果之后呢，首先第一就看一下白塞病这个蓝色的跟红色的健康对照是否可以很好的有区分开。如果能很好的区分开，就说明就是说我们这里面是有相应的标注分子的。那么第二呢，就是我们跟以往报道比较，我们发现跟以往报道我们检测到银屑病相关蛋白是以往报道的20倍，这也说明我们蛋白质组织它一个强大的临床的检测能力。通过对血液蛋白组的功能注释，我们也发现大量跟免疫性疾病相关的这些 biomarker 以及潜在的这些CirculatingProteome 临床治疗的一些靶点。那么这些是我们揭示白塞病它特性表达蛋白的一些分子机制、临床信号通路的信息。那么主要是以补体和凝血它的变化为主。那么红色是表达变化显著的蛋白，同时也是哪些蛋白是相当要靶，我们也进行了标注。那接下来就是关注到白塞病抑制的问题。首先我们把我们的得到蛋白进行了分子分型，我们分成了3类。我们看到3型它分布跟1型、2型是分得非常非常开的，1型、2型是比较接近的。那么从它的分子特征来看的话，就是1型它更注重于凝血，那么2型更注重于糖酵解，3型它更注重于炎症这个特征一块，这是不同亚型人群的分子特征。那么跟临床的分型在比较呢，我们看到蛋白质组分为3型，那么临床是分为2型。那么蛋白质组学1型、2型跟临床的1型是非常接近的。那么蛋白质组学3型跟临床的2型也同样是非常接近。同时我们通过样本的无偏向序列分析，我们同样验证了这一点那么就是说证明我们当时跟临床分析结果是一致性是非常好的。另外对于白塞不同的受力器官呢，我们它特应的表达蛋白和定位呢，我们又找到了一些蛋白可以定位到它的受力器官的特点，那么包括它的一个胃肠道的心血管受类的、心脏受类的、皮肤受类的包括这个关节炎受类的。那么右面是相应的表达蛋白的功能注释，它非常契合我们临床的它的分型的一个特点。那么通过这个结果就是说，我们进一步来找到就是与这个临床相关的一些标注分子，用 ELISA 方法再独立队列，又进行验证， 并且以临床的严重程度进行了相关分析。那么最终我们确定了3个与血管受累的标注分子，那么这个跟血管受累和非受累患者就有非常好的一个相关性。所以这个就是说针对血液蛋白质组开展一个临床分子分型的一个实例。最后再简要介绍说，这个对于临床治疗预测的，因为这个也是精神医学非常关键的一个内容。那么就是说，如何去改变当前的医疗模式，实现个体和群体进行个体化治疗、靶向治疗。所以这里面我们选择一个肿瘤的免疫治疗，因为它单独使用 PD-1 的有效率它是比较低的，所以我们鉴定一个标注，我们是可以提高临床的有效率的。那么针对这个呢，我们是首先也是呢，通过血液蛋白质组筛选了大量的血液中的6900它的自身抗体，然后鉴定了跟小细胞肺癌、跟肉瘤和淋巴瘤相关的这个治疗相关的标注。那么其中举淋巴瘤作为一个例子，那么跟治疗后3个月、4.5个月和这个6个月，我们可以看到，那么其中有一个标注分子是 EFE2， 它跟这个淋巴瘤啊，它验证的这个具有非常强的相关性。并且我们在临床2个队列也进一步验证，在 R 组中这个标注的分子它是非常显著性升高的那么再往下呢，就是说我们这个结果发现呢，就是说我们在患者体内，在10种癌症患者体内，我们发现这个癌症这个 PD-1 免疫治疗患者，它就存在很多天然的 PD-1 和 PD-L1 的自身抗体其中是这些抗体是以 IgE 和 IgG2 为主的，并且这些自身抗体它可以非常好的这个反映出临床的它的一个靶向治疗的有效性。下面就是说，而且是它对于总抗体来说它是没有差异的，但是它对于 IgE2 的患者，它可以预测20%的它的临床的靶向人群，它可以达到更好的疗效率。那么这个也是分析它的这个标注分子可以反映出肿瘤的靶向人群跟非靶向人群的一个抑制性的一个情况，并且这个标注我们也是在这个预后的能力对2个队列里面也进行了验证你发现这个 PD-1 和 PD-L1 它的自身抗体，它对于 PD-1 的免疫治疗也具有非常重要的价值。所以在这个里面，我们鉴定和验证了一系列有望用于免疫治疗的标注分子，所以这个我们也在往临床来进行推进。那么接下来呢，就是也是这个问题，我们不仅是说我们只是打造用的科研的整个的这个芯片，那目前呢，我们也在打造面向临床应用的免疫检查点针对一些自身抗体检测的临床应用型的这些蛋白芯片。那么这些芯片可以很好的检测我们临床它的一些 PD-1 免疫治疗免疫检查点相应的一些抗体药物分子，那么可以指控它们的这个特异性和交叉反应。那么最终呢，对于这个我们这个蛋白质组学研究成果，就是贺院士提出来的 PDBTM 应该落地，因为我们的这个最终的目的是说，研究成果不能待在实验室，要让更多的人要用起来，并且为人类造福。所以这里面其实它涉及的问题是非常非常复杂的一个问题。第一呢，就是说像这种转化研究投入是非常非常大的，所以现在就是说随着我们蛋白质组学这个大科学计划来推动，现在也是国家对这方面也是支持越来越大，并且现在资本现在也是对于蛋白质组学转化一块也是非常非常的重视。所以现在呢，就是通过引入大量的这个资本投入，会推进我们蛋白质组学转化的进程。那么同时呢，就是说我们做科研人员我们掌握了我们的优势，就是掌握了前沿的技术和高端设备，但是我们为了让我们的这个成果容易更快的更好落地，所以我们一定要跟临床专家和 IVD 企业进行互动，我们真正的了解临床和市场的需求。同时呢，我们建立成果的转化机制，真正的实现各展所长，各自有序。那么最后呢，还是我们要与监管系统也要互相沟通，因为我们是属于创新时期，一定要通过创新渠道和有力的政策来推动我们产品。因为尤其是对于癌症检测的问题，临床批证对于创新时期是非常非常难的，所以这里面我们一定要跟这个监管机构我们来沟通。那么最后呢，就是说对于市场和宣传教育也是非常非常重要的，能够让越来越多的人认识到，那么 PDBTM 在推动健康保障和经济发展中的一个重要作用。所以这个是这个多学科多部门联动推动蛋白质组学成果转化。那么最后呢，也简要的介绍一下，我们在2022年12月7号在 HuB 大会，贺院士向全球同行正式发起了人类蛋白质组学的导航计划。这个计划是非常宏大的一个计划，如果通过蛋白质组学来系统的解读人体蛋白质组构成原理和变化规律，实现蛋白质组学驱动精准学的范式改变，共创智慧医学的一个新时代。所以在这个里面，我们也非常欢迎国内外的专家和教授同行一起来加入到我们蛋白质组学的这个大的家庭里面，我们一起来共创智慧医学的一些新辉煌。那么最后呢，谢谢大家。丽琴
，OK，thank you。
建南。
那是用中文吧。那个对于小波教授的这个有没有什么问题啊，各位老师？然后如果我们可以待会儿跟约翰一起用English 做一些 panel discussion。Maybe we can discuss your research together with Johan during the panel discussion.
Okay, yeah, we can use English. Yeah, it's fine.
Hey, Johan, can you understand? But I believe you know what. You are muted. You are muted.
I read from the slides that
you can read
international language.
So the figures, the data,
Yeah.
no language. Basically, he shows he's a very cool technology and applied to multiple diseases and develop a kit which can be used in clinic. And this is the main goal of our research. I think this is also consistent with what you have said just now. There are so many things, so many technical details to deal with. Sample collection, sampling, the data acquisition using mass spec or O-link or whatever other technology, arrays like what Xiaobo and Shense are using, and the data analysis, standardization, and so on. All these have to be discussed before we can bring this to clinic, to the people at home and the clinic. So any questions, comments to those two talks? So
hi. So we can have discussion now, right? For
yeah, go ahead.
The question was interesting. Yeah. Hi, Johan and Shengce, Tianan, Yingjiang. So since there have kind of different technologies, for the proteomics, So, but we are facing one important issue is whether we can make the same achievement as genomics. So, do you think what kind of issues still exist behind our proteomics technology so that we can push forward for our proteomics and precision medicine?
I mean, people call proteomics or proteins the intermediate phenotype, which means that they are an active component which changes. So I think proteomics is a monitoring technique. It's not a one-stop shop for doing things once. I think you need to create a movie where genetics, will give you your lifetime risk, but proteomics will tell you where are you on the risk scale right now, right? So I think that's what the important component is that we need to make people understand, I think. And
okay.
It's a reactive phenotype, so treatment effects, all these things. I think it's going to be very interesting.
Yeah. Great.
Answer Xiaobo's question, I think for proteomics is the problem is too dynamic. And when we are dealing with clinical sample, try to identify some like a reliable biomarker. So another problem is from people to people variation. That's give you a big noise. So that's why we always see there's a lot of paper come out say we have potential biomarkers, but eventually only a very few can eventually go to clinic, right? That's a problem. I think the simple here is one thing to really make proteomics works in clinical settings. I think the standardization is, maybe the most important part. How to make everybody can achieve the same set of results, use the same set of sample. So I think that's one question somebody asked Johan, right? Back to your talk. So that's very important. We have to start with that. Then sometimes we can get some very reliable and robust biomarker. And based on that, I think we can really get something which is very useful, right? Can I?
So I have a technical question. the blood is a very complex sample in terms of protein analysis. And one can measure by mass spec and by depletion followed by mass spec, like a SEER or many other people doing using antibody to deplete or reduce the abundance of some high abundance protein. And can measure by Olink and by what is Schaboth method, synthesis method. There's so many methods, and there are so many measurements, so many type of readouts. They have different range, different numbers. But in reality, every protein in a particular sample, they only have one concentration. So actually, we are all deal with relative quantification. And some people saying they are doing absolute quantification by building calibration code. But maybe very few people doing that. So when I talk to some people from the authority, healthcare authority, want to commercialize some analysis using clinic, and they will ask me, do you have the exact concentration of this particular protein or whatever peptide in the blood? Then I cannot answer that. When we use mass spec, the proteins are digested into peptides. We measure peptides. Unless you synthesize the protein with some label, then you can know the absolute quantification. But for people asking this question, they are dealing with the small molecules like some vitamin, because vitamin seems like can be precisely measured. And some people are talking about measured RNAs. They think the RNA can be precisely measured with their qPCR. There are a lot of quantitative approach. But so I want to have your comments. Do you think protein can potentially measured precisely by whatever method that you are using? I mean, every one of you are using maybe a different or a slightly different approach. Do you think your method can precisely measure the concentration of a particular protein in a particular sample? Maybe your turn. How about you? You have tried different methods.
Yeah, I mean, of course, I know the roots of, producing a standard, making Edman degradation and all these things. But I agree, with you, if you have only a sub-representative of a protein, meaning you just have a couple of peptides, and then I think you can do this on a peptide level. And I guess the larger the protein is, the more difficult it will become. So I think if we can start, if we could start with smaller proteins, and see, is it actually technically feasible? How feasible is it? I think I haven't really, seen that come to real life. I know people, doing targeted approaches, but it's still rare, I think.
Yeah, we are doing some targeted approach. Now we can develop one or two very robust peptides for a particular protein by ignoring many other proteins of similar sequences, isoforms, splice invariant, modified, glycosylated. We ignore them. Only use the database of 20,000 proteins, and we assume we can measure those peptides precisely. But from protein digestion into peptide, there's still a lot of variabilities. And the peptides go through the chromatography and ionized. There are also a lot of variabilities. So my preliminary thought probably is not technically feasible to determine a protein's exact concentration by bottom-up mass spectrometry. Don't know what other people think. So this is actually this is a critical question if you want to move to clinic. If it is measurable, then of course you need to provide evidence. If it's technical not feasible, you will never be able to pass the standards set up by the healthcare authority. They have the mindset for small molecules and RNA or DNAs. So what do you think? Do you think your array can measure a protein precisely to determine the absolute quantification?
I don't think so. It depends, it depends, because there are a variety of different proteins in the circulation system, also in the tissue. I believe I believe the proteomics will be different from genomics now. So there are too many different types, just like proteins, protein post PTMS, truncations, complexities. I think so for the proteomic technology, it depends on the which type of targets you are going to detect. So just focus on the specific type, you probably can develop the absolute quantification method. That's what I believe. But if we want to measure all of the proteins, that would be a big challenge.
Okay, so there are two questions. So the first question is, if you have one protein of interest, that's just one protein, not about the other proteins, do you think you can precisely determine its absolute concentration in a sample?
Yes, yeah, this question, yes, yeah.
How?
Yeah, for one proteins, there are many different options, just like we can use ELISA, we can use chemical luminescence, we can use kind of POCT, so we can use very high throughput and cost-effective platform to quantify one protein. But if you have 3 or more proteins, that would be good to use different platforms such as Luminex, so Johan is very good at Luminex technology. Also the MSD, also the Simoa. So there are several options for the several proteins, just like 3 to 10. But if the protein number is more than 10, that would be different thing. So for to detect their concentrations, it depends.
Yeah. So how about the center? What do you think?
For this question, I think one way is to use antibody microarray. For antibody microarray, if you have like a set of good quality antibody, then maybe you can like measure tens to hundreds of proteins simultaneously to give some quantitation like information. That's one way. Another way, I think maybe there's another choice is O-link. O-link, if you combine, you use mass spec to identify the biomarker, then you develop something which is similar to O-link, then maybe you can also do some like very precise quantification. I think that's my answer. I think Johan can say something about this, right, Johan? Since you are leaving in 5 minutes.
Yeah, I think what we have done, I think we've actually compared a couple of different methods measuring the same protein and actually using different antibodies for the different methods.
Right.
But it's funny, even though the data correlated very nicely, the difference was the standard. Because the standard, how the standard was quantified made the curves, move in parallel. So I think, in that sense, if you have a combination of techniques that are equally well recognizing the same standard, I think you could probably do it as a combinatorial investigation. But nobody will pay. Okay, you measure one protein, but you need 5 tests to do this.
Right.
So I think that's probably a possibility. Yeah.
Okay.
So Dr. Chen, you have some comments?
是的，不好意思，我就用中文吧，因为刚才听了小波老师中文讲的那些内容，实际上我们在这个刚才也提到了，这个可以用什么平台来转化这些内容。那么我们实际上是有蛮多平台的，从Mary 的角度来说，我们有这个光学的平台，也就是生化的这个部分，还有一些免疫的蛋白的平台。那么相对于蛋白质组学和基因组学来说，实际上蛋白质组学我们认为从我们的这个时间来看更容易被临床接受，因为一个方面是从价格的维度来说，它的成本肯定是要低廉的，那么相对基因组学来说，另外就是方法学来说它会更成熟，所以更多的这个蛋白质的一些标志物，包括肿瘤细胞肿标的早期的识别，肿瘤的一些标志物，那么刚才小波老师提到的一些大的小的蛋白分子的标志物，实际上都可以在这个已经成熟的这些平台上来转换。当然我们也愿意跟各位科学家一起来做这个事情。 对，这就是我想补充的一个点，谢谢 ，Thank you。
Thank you. So Dr. Chen said he's willing to help to translate the proteomic findings into commercial products. So actually my point was, if it's so difficult to determine the absolute concentration of the proteins, probably it's not necessary if the goal is to perform diagnosis or monitor disease progress, because relative quantification with good standards probably is sufficient. If this is the consensus from our community, probably from your HuB human plasma project, then we can write something together, write some opinion, a commentary article. Then we can hand this to the healthcare authority in different countries. Say, we don't have to really determine the absolute quantification. We can already develop a product to monitor COVID, monitor many other diseases. And this is different. It's biology. It's different from metabolites and different from DNA, RNA. And this is protein. They are different. So we need to educate them. This is a different thing. Otherwise, we'll be trapped in how to prove you have the absolute concentration of every protein.
Yeah, I think it's a very good comment. And sometimes when I explain my work to people, I mean, I use the metaphor of going to the movies. Let's say proteomics is like going to the movies. It starts at some point, and then you have the experience, and it ends. And the things that happen in the movies, it doesn't matter if you sit in the first row or in the last row. It's your experience with the movie, right? So, and that's the change that you will experience in, let's say, you come to a clinic, okay, let's get part of this testing program, which means you get repetitive sampling, and then you get not the picture, but you get a whole movie about your health. And then the study ends again, then we do it again. So next time when the Star Wars #2 comes and so forth. So I think it's this dynamic that I think we need to see. And I fully agree that it's maybe proteomics is not really analytical chemistry. It's more analytical biology. Right? So okay, I unfortunately need to leave. But it was a great pleasure for me, and thank you. And hopefully, if there are questions, please reach out on whatever platform. Thank you very much.
Okay,
Thank
thank
you.
you.
Thank
Okay.
you. Thank you.
Bye-bye.
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 Right. Bye.
Bye-bye.
Bye bye.
Zhang Yin, do you have any comments?
中文吧。
张老师有没有什么comment？
哦，我刚才静音了。其实，咱们刚才讨论的是那个转化的那个问题啊。但是其实我觉得从蛋白质组做发现的这个地方，其实就蛮有挑战的。但是就是如果我们真正的能够发现一个比较好的标注，其实转化其实我觉得应该刚才那个杨老师他们说的，其实还是可以的。就是我觉得就是说如何从我们的这个样本到后面能够发现一个非常 soluble 的一个 biomarker， 这是我觉得是最有挑战的地方。因为现在蛋白质组学这个技术，我不知道现在 Olink 怎么样，就是它这个蛋白质组学这个技术，一个是对这个检测的灵敏度，还有它的这个刚才讲到的这个人跟人之间的这个 variation 的这个变异，怎么去发现？你会找到一堆差异蛋白，但谁会成为最终的真正的 biomarker？ 这个我觉得是我现在觉得最大的挑战。但一旦我们确定了这个哪个蛋白是了，那后面转化，我觉得他们肯定有办法。对他们工业界的来说，他找好用的抗体开发什么的。
有时候您在实验室里面，比如找到一个 marker， 好像是很好，那一转化之后可能就不一样了。
对，是的，就是实际上这个技术和技术之间这个 gap， 还有不同的技术可能针对它的这个定量的质谱的是肽段呀，还有这个免疫组化，可能针对的它的这个 ELISA， 它可能针对抗体的不同的表位，这可能都是之间的这个将来能不能转化成功的一个关键。
对，我知道 mass spec 呢，因为我们通常 discover 的时候是用DDA、DI 这种 discovery proteome， 但是转化的时候那只能用 targeted proteomics 所以它的ionization、fragmentation 都可能是不一样的，所以整个它会有一个 gap。刚才小波那个方法，可能芯片的方法是不是相对简单一点？然后这个 群里面还有Olink 的伪经理 Patrick， 他说这个 Olink 可以私人定制啊，就是定制某些
这个如果说 Olink 买单的话，我们就找他定制。
Olink 太贵了，现在对我们都用不起，更何况临床了。
而且 Olink
对 Olink 让大规模用的话，还要把价格要降下来，因为现在确实是一般的实验室可能还是承受不了这个价格，尤其是定制化可能会价格
做不了发现的，基本上就是知道什么东西再去做，可能更好一点哈。
对对对，这样是比较好，
比较明确的。
所以 Olink 应该赞助一下我们这个会议，这样我们更多的讨论，更多的好的，刚才那个杨晨博士也讲的蛮好的，我觉得小波你跟麦瑞做的那个试剂盒，对应做成试剂盒了，我觉得非常非常不错。这个
对，其实这个里面就是我感觉这个里面确实从质谱来讲的话，就是还是要可能还要急需解决一个，就是说，因为现在毕竟质谱在临床的话，好像蛋白质谱还没有直接的这个临床的一个平台，所以说现在可能来说，基于免疫法，可能相对于质谱来说还是比较容易去接受的，就是临床转化的，但是它就会存在一个 gap， 就是刚才就是您这边提到的，就是质谱检测是太断。但是说免疫法它检测是蛋白，这样的话就是说，第一个方法学可能会有一些不一样的东西，就是质谱检测的效果非常好，但是从这个免疫法的话，它可能会有可能检测的效果没那么好，这个有可能会是一个 gap。但是如果说我们标注足够多的话，可能从里面挑一挑，可能还是会有一些比较好的一个标注在里
面。是的，我也补充一下，小博老师说的就是。我们在肿瘤的早期筛查，就是在肿标这一块，包括在一些激素方面，就是我们还是致力于找到更多的这个标志物。那么特别是能在现有的这个我们的生化免疫平台上用的这些标志物，我们都愿意跟大家一起，包括来支持大家，来跟大家一起来合作，来把这些标志物做一个转化。对，这也是我们未来发展的一个方向吧。
对，那个杨老师，那个我顺着您这个话来问一下啊，就是您从企业来讲，您认为标注做到什么程度，您企业这边就比较愿意去接手？哪管就是说，我们可以双方共担风险嘛，对吧？我们选择这个1、2个这种 topic， 然后我们实验室可以现在推到一定程度，这个时候，你们企业就愿意来共担风险来做这个事儿，对不对？您感觉这个 time point 大概是一个什么样的一个节点会比较好呢？
我觉得主要还是就是您说的，就是性能，就是我们日常所关注的这个精密度、准确性，那么包括线性、重复性这些核心的性能，实际上也就是您说的这个注册这个体系所关注的。另外，我们特别关注的是临床价值，就是哪怕那天其实之前在交流的时候，跟小博老师也提到说，哪怕一些比较前沿的这个新的标注，我们目前没有看到它特别大的市场，但是它的临床价值是显著的，特别是一些这个新的一些具有诊断意义或者临床价值，能够解决临床痛点和临床问题的这样一些标志物，是我们特别关心和关注的。另外我们也能够这个特别在血清学的这个相关的标注，可以用我们这个平台来做一些临床验证。那么特别在性能上的这个维度，就是我们所关注的这个标志物的系列的检测的性能，如果性能是好的，然后具有很大的这个临床意义和临床价值，我想这个我们也是特别愿意的，从企业的这个角度来看，大概是这样子。
迈瑞还是很大的一个企业，可能不知道是不是每个人都知道。那个，要不杨博士讲一下你迈瑞主要做什么事情？我记得是做医疗这个仪器的哈。
是的，
检测方法吗？
我给大家介绍一下吧，就简单的介绍一下。迈瑞现在就在全国的医疗器械应该是 top 1的位置那么医疗器械当然就包括了体外诊断，包括了生命信息，所谓的生命信息就是呼吸、监护、麻醉这部分，还有我们的影像诊断。影像诊断我们这边主要是超声。那么这些加起来，在整个医疗器械的 这个行业里面，目前迈瑞是第一的。在全世界大概能排到32，今年应该是32的位置。那么也有榜单说排到了27、8，就在整个世界的医疗公司来看。那么在体外诊断这一块，也跟大家汇报一下，我们也主要有几个平台，一个是这个细胞学，就血液细胞学的这个平台，主要是流式细胞术，那么是对血液细胞的分析这样一个平台。还有就是我们常见的这个生化分析的平台，就是临床生化分析的平台，临床免疫分析的平台。当然我们也在看这个一些新的方向包括质谱，包括测序的方向。但是迈瑞目前还是以传统的这个业务为主。那么几个业务来说血液细胞检测的这个业务，我们目前是中国第一，就是加上进口的公司，包括可能大家了解日本的西斯美康，它过去是在中国第一，我们现在已经超过它2年了，是中国第一。然后在生化检测，就是常规的这个肝功能、肾功能血脂类的这个临床的这个检测上我们是中国第一，也超过了贝克曼和罗氏。那么免疫到去年，也就是2022年的时候，我们应该排到了中国第4，前面是罗氏、贝克曼、雅培，我们超过了西门子，目前是中国第4。大概是这几个常规的平台。当然新的方向包括 PCR， 包括测序和分子，我们正在看，但还不是迈瑞目前的主营的方向。所以我们还是强调说，在这个临床上所使用的这个常见的和传统的标志物这块，我们应该还是有一些优势，也能提供相应的检测平台大概是这么样一个情况跟大家汇报。
好的，非常好。现在大家都在谈科技自立自强医疗器械有迈瑞这样的企业，还是就可以有一些不要什么都从国外进口我就回答那个网上有一些问题有个问题是说多肽是否可以作为标记物？多肽当然是可以作为标记物，用质谱去测的都是大部分都是多肽，应该说99.9%以上都是多肽。然后有一个问题说，如果是很低丰度的多肽 low abundance how to enrich or how to capture？ 那这个据我的经验呢，就需要通过一些抗体的富集可能要么先富集蛋白要么就去列高丰度蛋白，要么就用一些 beads像 nano beads 可以去 enrich 一些 peptide， 包括phosphoglycopeptide 等等。那个时间过得很快了，现在已经快9点50了所以今天这个 meeting 我觉得还是蛮有意思的。但我还有一个很大的问题呢，没有回答，但是我想可能要等下次了，因为这个 title 是循环蛋白质组学循环蛋白质组学除了血液之外，还有其他的。
对，尿液。
我想问这个问题，今天可能没时间了，我们可能还要，因为这个 topic 非常的 interesting， 也并且是找 biomarker 特别好的一个这种 potential。我刚才还有好多人私信我，在期间也讨论一些问题。我觉得我们以后可能还可以办一个series 2，然后专门讨论这个 CirculatingProteome， 还有好多可以讨论的地方。我今天非常感谢这个江英老师、申策、小波，还有他的名字怎么读啊？Johan
Shrink，就是标准的这个读法。Johan，
我以前见过他，但是没有太多的。
你要揪起来有点像中国人的名字了哈。对，
我觉得讲的蛮好的，这个他的就非常系统。你看他做那个 micro sampling， 那么小一点点，然后放在家里。
那个非常好。
中国有没有人做这个事情？中
听说
国小波，
好像我还没有听说过国内有人做这个。其实那个东西看起来小，其实特别有用，特别有用。
对，它非常小并且信奉给寄到实验室来。
对。
但我有点怀疑它的稳定性。它那个就算是干的，用抗体去测肯定不够稳定，用蛋白。
对，干的看他做什么，估计他那个 paper 上肯定是做过某种修饰。因为我大概几年前去过一次澳大利亚，当时有一个人拿一个东西说他那个就是特别稳定，但是呢，他体积不能控制。他现在这个可以控制体积，如果再加上稳定性，那如果说实验室收集样本就特别好，就收了先放了，然后最后再测嘛，它一致性非常好。对。
这些迈 也可以考虑一下。好，
嗯，
那我就今天就
好的
先这样吧。
好的
各位老师，
好
感谢各位参会和 Doctor。好，谢谢。
好的
好的，
好的，谢谢天南
哎，
啊
再见，
生顺
好
肖老师杨老师啊，好再见啊，
哎，谢
谢谢丽
谢各位老
群，
师，谢谢各位老师，好，拜拜。
拜拜。
各位老师，拜拜。